Hello friends, welcome to the today's session on separation techniques 4. So in this session we will be discussing about separation of proteins by using isoelectrofocusing as well as 2D gel electrophoresis. In addition to that, we will be focusing more about the separation of nucleic acids by using electrophoresis. In our previous sessions we have discussed about the separation of proteins by using page, SDS page and paper electrophoresis. Let us begin the session. In this session, we will explore more on the fundamental principles and applications of isoelectrofocusing, two-dimensional electrophoresis and Acros Gel Electrophoresis. Along with that, we will be focusing about the applications of electrophoresis. In our previous session, that is separation techniques 3, we have discussed about the separation of proteins based on the SDS space and as well as the paper electrophoresis. So here IEF stands for isoelectrofocusing. That means we are going to mainly focus on the isoelectric point of the proteins. Coming to the history of this technique invented by H. Svensson, a Swedish scientist. Here this technique is having an advantage of, that is, high resolution power. We will later see what does exactly mean by the high resolution power of this IEF. Coming to the principle of this electrophoresis, that is IEF, is separation of proteins achieved based on the zwitterion state, that is the isoelectric point. Hence we call it as isoelectrofocusing. So if you recall the structure of proteins and the composition of proteins are made up of amino acids, and there are various types of amino acids like acidic, basic and the charged, uncharged ones. So when you dissolve these proteins in a suitable buffer, they will obtain a particular charge. Here similarly, based on the pH also, these proteins will attain a state that is called zwitterion state or isoelectric point. At this state, proteins doesn't have any what is called particular specific charge, but they have both the positive and as well as negative charge. That's why they will remain what is called as stable at this particular point. That is called isoelectric point. That means electrically they are stable and they have both the positive and negative charges here. That's why this ion is called as zwitter ion. So as we have discussed about this SDS page in the previous sessions, so this in SDS page we are going to prepare the gel on our own. But whereas to perform this IEF, there is no need to prepare the gel material, but there is commercially available strips are there, which we call them are IEF strips, and these strips are ranging with different pH gradients. Like you can see here, these IEF strips are ranging from a pH with various ranges, and the sizes are also like 7 to 11 centimeters range. Here in this image you can see the representation of how to perform the isoelectric focusing. So if you see this image, the middle one is nothing but the IEF strip, and on the top side you can see a cathode, and at the bottom that is anode. That means we are going to connect this strip in this direction, like one side is towards cathode, the other side is towards anode. And moreover, if you observe the right side here, we have mentioned about high pH and the bottom is low pH. That means here the pH on this strip is ranging from high to low pH. Okay, and low pH is towards anode, whereas high pH is towards cathode. Okay, on the left side of the image, if you observe, so here the top one is saying that sample introduced at pH above the isoelectric point. So as we just now said, the major principle working behind this technique is isoelectrofocusing. So to perform this isoelectrofocusing, we need to have a range of pH from high to low. And here we are going to introduce the sample at both the high pH and as well as the low pH. If you observe on the strip, we have mentioned here that is sample introduced at pH above isoelectric point, and the bottom also you can see sample introduced at below isoelectric point. Okay, and on the right side you can see the direction of migration of anions and the direction of migration of cations, and in the middle there is a state which is called as zwitterion formation, and here you can see these zwitterions are immobilized. Okay, It means here what we are trying to convey you is, so when performing this isoelectrofocusing, we are going to introduce the sample towards the low pH and as well as the high pH. And for example, at low pH, if you see low pH, there are, because of more protons, these proteins will get a charge of positive charge. And as we know, the positive ions generally move towards cathode. Okay, that's why on the right side we are showing the direction migration of cations. That means they are moving towards, these positive ions will move towards the cathode. Similarly, when you introduce the protein mixture at high pH, the proteins will get net negative charge. Okay, and when you run the electricity, when you allow the current to flow through this strip, what happens? These negative ions, that is the anions, will move towards the anode. But because of there is a range of pH from high to low, both the proteins, according to their pH and isoelectric points, the moment they attain the state of called Zwitter ion, their mobility is going to be arrested. At that point, we are going to immobilize the sample components at their isoelectric point. That means here we want to say that separation is achieved based on this isoelectric point. In the middle, if you see, these bands, they are distinguished bands of the distinguished samples. So in this slide, we are trying to explain you with a colorful image that how the separation is going to be achieved. So if you see on the left side of the image, there is a stable pH gradient which is again ranging from, if you see, 0 to 14, as we said, high to low. Okay, towards cathode it is high and towards anode it is low. Okay, and the molecule, when it is introduced at low pH, it is going to have a positive charge, and the molecule that is introduced at high pH, it is going to have net negative charge. Okay, and in the middle one, the middle picture shows here, when you allow the current to go through the IEF strip, what happens? You can see here the positive ions, that means from towards anode, they are moving towards the cathode topwards, whereas the negative ions from the top, they are moving towards down. These arrows shows here the movement of the ions from towards opposite directions from their native places. In the third image, you can see all these ions, they are now separated. If you see, especially the green one, see the green one which is coming from actually towards the high pH side. In the first picture you can see this is the green one which is moved, now this is here. That means this particular protein or ion, you can say, is having a isoelectric point which is near to 5. You can see here. Even the table shows here the green one is having a isoelectric point pH 5.6, and similarly the red one is having somewhere around 7. The yellow one is having somewhere around in between 8 to 9. So like this, the ions which are charged and they are separated, but their separation is based on their isoelectric point. So if you observe these three images and compare them, you can notice that initially they were either positively charged or negatively charged. As the IEF is progressive, the ions get separated, but separated based on their isoelectric point. In the third image, as you have observed, the separation occurred, but occurred based on their isoelectric point, not that whether you have introduced them towards the anode or towards the cathode. Irrespective of that, they get separated based on that isoelectric point. This is the versatility of this isoelectrofocusing technique. Yeah. So in this image, you can see how the apparatus looks like. So in this is the apparatus which we use to perform the isoelectric focusing. On the right side, you can see the IEF strips. Just now I mentioned that. So here there is no need to prepare the isoelectric strips manually. Commercially they are available in the market. One can buy and load the sample as per the instructions. And here on the bottom side you can see the direction of migration. The moment you load the sample and run the electrophoresis apparatus, so here what is happening, the direction of flow, you can see the tracking die which is moving towards bottom. If you go here, you can see the last one, the die is reached the bottom of the strip, and here you can see the proteins which are gray bands get separated. they are getting separated. That means as the IEF is progressing, the proteins are getting separated based on their IEF values. Okay. So now we will know something about the application part of the IEF. So as we have discussed about the applications of SDS page in our previous sessions, now we will discuss something about the application part of this IEF. So here this IEF is responsible for the separation of mixture of proteins into individual proteins. So if you are taking any tissue and subjecting it for homolysis, and the homogenate which you have obtained, that means the crude homogenate, you can subject for the IEF, and from that you can separate the individual proteins. That means crude can be separated into individual proteins. And similarly here, the other property of this IEF is, the important application is its high resolution power. So in the very beginning we said about the resolution power. That means the resolution power is, this IEF technique can separate your plasma proteins into individual 40 bands. Whereas if you compare this IEF with that of the paper electrophoresis, in paper electrophoresis we found that the serum proteins we are going to get 5 bands. Okay, but whereas in IEF the same protein you can get separated into 40 individual proteins or 40 individual bands. That means IEF is having a high resolution power when compared to the other, like your native page, SDS page, and as well as the paper electrophoresis. That's why to separate the crude protein mixtures we are going to depend on this IEF technique. So moving further, let us discuss something about two-dimensional gel electrophoresis. So here the name itself indicates that is 2 dimensions. There are 2 dimensions. So you can see in other words, that is, one is horizontal, the other one is vertical, or it is nothing but the combination of 2 techniques. So that is, one is your IEF, the other one is SDS space. When you combine both the IEF and as well as SDS space, the product is nothing but your 2-dimensional electrophoresis. That means in one IEF you are going to separate the proteins based on their isoelectric point, whereas in the case of SDS you are going to separate the proteins based on their charges and as well as the sizes. Let us explore more about the 2-dimensional electrophoresis. So as we said, this is IEF is nothing but the horizontal one, and whereas the SDS one is nothing but the vertical mode. That's why by combining the horizontal vertical, you can say, otherwise IEF and SDS, this technique is known as 2-dimensional electrophoresis. Okay, and then so here first of all we are going to take the protein sample and load on the IEF strip, and then we are going to subject for the IEF, that is isoelectrofocusing, where proteins get separated based on their isoelectric point. Later on, the same strip of IEF is transferred over to the SDS gel, where again separation occur but based on the molecular weight of the proteins. In first case it is based on the isoelectric point. In this second step it is going to separate based on the molecular weight of the protein. So you can see the flow of what is called as this 2-dimensional electrophoresis. On the top you can see the IEF strip, where we are going to perform the load the sample here on the left side, and we are going to perform the IEF. So by this IEF, what is happening? The proteins are getting separated based on their isoelectric point. After that, we are going to take that strip and loading it on the SDS. You can see in the bottom rightmost corner you can find the SDS gel with the IEF strip on the top. Now this movement is towards the vertical direction, whereas the first one is towards the horizontal, this one is towards the vertical, and these spots which you are able to see here on this picture are nothing but the individual proteins which have been separated by using this 2-dimensional electrophoresis. Here you can see in this image how the apparatus looks like. Left side you can see the entire apparatus, which is more or less similar to that of your SDS page, but whereas the size and the power will be varying from the SDS space to that of the 2DS space. On the right side you can see the gel which is subjected for the SDS space. So on the top side we are going to keep the IEF strip and allowing it to go for the 2-dimensional electrophoresis. So on these spots, these are the spots which are of separated individual proteins. So the major application of this 2-dimensional electrophoresis is, it is useful to separate a mixture of proteins, and you can see here a mixture which is containing almost all 5000 proteins can be separated by using this two-dimensional electrophoresis. And the major advantage of this 2D is you can separate crude protein samples which we obtain from the either from plasma or serum or from the tissue homogenate. You can subject those proteins to get separated into individual proteins by performing this two-dimensional electrophoresis. Yeah. So far friends, we have discussed about the IEF and the two-dimensional electrophoresis. Now let us have a brief look on the Acros Gel Electrophoresis. In this Acros Gel Electrophoresis, so the major thing is we are going to use the agarose is one of the major important ingredient in this gel, which we obtain from the seaweeds. And generally these agarose gels, depending on the nature of separation, we will be using the concentration of 1% to 3% based on the purpose of your separation. Okay, and the same time here the gel is prepared in such a way that the range is from low concentration to high concentration. So whereas low concentration will provide you the larger pore size, whereas the high concentration will provide you the smaller pore size. For example, if you are separating a DNA or anything which is of bigger in size, then you will be using the big pores size, whereas if you are using small molecule, then you are going to use a thick gel. Okay. Yeah. So in this image you can see here how the Acros Gel Electrophoresis apparatus looks like. On the left side you can see which is also having a buffer tank. The broader one is a buffer tank, and where we are going to load the buffer, and in the middle there is a casting tray. In this casting tray we are going to pour the gel. Once the gel get solidified, we are going to remove the comb, and into that wells we are going to load the DNA sample to be separated. And the moment you allow the current to flow through these electrodes connected to the buffer junction, the DNA sample gets separated. On the right side you can see the apparatus and the setup, how it looks like. The same thing as in the case of your page also, we have a power pack and as well as electrophoretic system. But here this electrophoretic system is nothing but a submerged or submarine electrophoretic system. Why? Because in this Acros Gel Electrophoresis, the gel is completely submerged inside the buffer. That's why this is called as submarine gel electrophoresis. Yeah, let us see the brief procedure how to perform the Acros Gel Electrophoresis. In this also, once you prepare the gel, so before going to cast the gel in the casting tray, we are going to add this fluorescent dye, that is ethidium bromide, and then we are going to concentrate the DNA sample by using 50% glycerol, which will enable the DNA sample to retain inside the wells. And then one more point to be remembered whenever you are handling this Acros Gel Electrophoresis, there is a precaution to be taken care. That is, do not touch this ethidium bromide with your naked hand, since this ethidium bromide is a carcinogenic agent which may lead to cancer. So that's why always better avoid the touching of this ethidium bromide through your naked hands, and wear surgical gloves and goggles to avoid the exposure to the chemicals and as well as to the UV radiation. So in this slide you can see how the gels of Acros Gel Electrophoresis appear after separation. Left side if you see, these are the gel bands. Here you can see the bright illuminating gel bands. So this brightness is because of the presence of the ethidium bromide, and this dark band, this dark gel here, it is obtained under gel documentation system. So this gel documentation system is the one which helps you to capture the good resolution images of the gels of the DNAs which we are going to separate by using this Acros Gel Electrophoresis. And the brightness is because of the presence of the fluorescent compound, that is ethidium bromide. On the top side of the gel you can notice the presence of wells here, and these are the wells where we are going to load the DNA sample. In these wells, here also you can see the wells. Okay, on the right side gel is the one which we obtained under UV transilluminator. That means For visualization purpose, you have two options. One thing is you can use the gel documentation system. If you want to capture the image and measure the intensity of the gels and wells, you can go for the gel documentation system, where you can free from the all radiations. In other case, that is UV illumination. In this purpose, we are going to use a UV transilluminator. Here we are going to put the gel on the UV transilluminator and close the lid, and once you put on the UV light, the gel bands will appear like a bright orange color, which says that these bright bands will indicate the presence of the DNA inside the sample. These bright bands, they are indicating the presence of the DNA sample. So coming to the application part of this electrophoresis, now we will be covering the applications of all electrophoresis, ranging from paper electrophoresis, Acros Gel Electrophoresis, and page, SDS page, IEF, 2D. So we will cover all the applications of all types of various electrophoresis techniques so far we have discussed. As we said, this electrophoresis is having major applications in separations, especially in studies of medicine, clinical and pharmaceutical research. And apart from that, the paper electrophoresis is an essential technique which we use in clinical laboratories to separate the proteins and isoenzymes and hemoglobins. And PACE is the one which is useful for the separation of small fragments of nucleic acids. And as we said, SDS is useful for the separation of proteins based on the size. Here in this graph, we are trying to explain you how we can measure the molecular weight of a protein by plotting a graph against molecular weight and the mobility. If you see, higher the molecular weight, lesser is the mobility. For example, only 0.2 mobility. 0.4 is again 8. So the molecular weight, as it is decreasing, the mobility is going to be increased. Similarly, in addition to this PACE, SDS page and acetate electrophoresis, we have another variation that is called PFGE, pulse field gel electrophoresis, which is useful for separation of chromosomal, genome and sequencing studies. And in addition to that, these electrophoresis techniques can be used for performing what is called as DNA fingerprinting and DNA restriction map development, which will enable you to know the genetic analysis. And similarly, electrophoresis is a major technique which plays a crucial role in the blotting techniques. So friends, let's sum up. So in this whole session of separation techniques, we have covered from separation techniques 1, 2, 3, 4. So in the first one, we have covered about the basics and centrifugation technique. In the second one, we have discussed about the chromatography. And in the third one, we have discussed about the electrophoresis of proteins. And the last one, we have discussed about the proteins and nucleic acids. Okay friends, so far we have discussed about various types of separation techniques which we will be using widely in the biochemistry for the purpose of separation and analysis. So okay friends, thank you one and all.